Today we'll talk about the types of RNA. What are the types of RNA? There are different types of RNA. For example, we have messenger RNA, mRNA. We have transfer RNA, tRNA. We have ribosomal RNA, rRNA. We have small nuclear RNA. We have cytoplasmic RNA. Another type is small nucleolar RNA, then microRNA. Small nuclear RNA and then microRNA. Another type is heterogeneous nuclear RNA. Heterogeneous nuclear RNA. So these are the various types of the RNA which are present in a cell, and we will discuss them one by one. Among these, in case of prokaryotes, there are three types of RNAs. One is messenger RNA, another is transfer RNA, and third is ribosomal RNA. These three types of RNAs exist in prokaryotic cells. So we have prokaryotic cells. In prokaryotic cell, there are three types of RNAs. One is messenger RNA, transfer RNA, and ribosomal RNA. While as in case of eukaryotes, we have, in addition to messenger, transfer RNA and ribosomal RNA, we have different types of RNAs. For example, small cytoplasmic RNA, small nuclear RNA, small nuclear RNA, heterogeneous nuclear RNA, microRNA. Today we will discuss about one of the important types of RNA, that is messenger RNA. So this is we are going to discuss messenger RNA. Messenger RNA. Messenger RNA, mRNA. The term messenger RNA was for the first time coined by Jacob and Monod. This term messenger RNA, it was given by Jacob and Monod. This messenger RNA, it carries the genetic information in a coded form from DNA to ribosomes. We can say that for protein synthesis. This mRNA, it carries the genetic information in a coded form from DNA to ribosomes for the protein synthesis. So it carries message. This messenger RNA, why it is called as messenger RNA? Because of the fact that this messenger RNA is called as messenger RNA because it carries the message, it carries the instructions from DNA to ribosomes for protein synthesis. So it is called as messenger RNA. It carries instructions, or we can say it carries message in a coded form. Message in coded form from DNA to ribosomes for protein synthesis. This Jacob and Monod, he for the first time said that these mRNAs are the informational molecules. These are informational molecules. informational molecules. This Jacob and Monod said that these mRNAs are the informational molecules, as they carry the information, they carry the message, they carry the instructions in a coded form from the DNA to ribosomes for the protein synthesis. For the protein synthesis, these are called as informational molecules. This mRNA, mRNA is the longest RNA. mRNA is the longest RNA. Yes, mRNA is the longest RNA. This mRNA is the longest RNA, and this mRNA constitutes about 5% of total RNA content of the cell, total cellular RNA. It forms approximately 5% of total cellular RNA. What I mean to say is that if there is 100%, we can say that there is RNA, 5% is contributed by messenger RNA, mRNA. So approximately 5% of mRNA, 5% of RNA present in the cell is mRNA. This mRNA has short lifespan. It has another important point. It has short lifespan. We are discussing it with the messenger RNA. So messenger RNA is the longest RNA. This it is approximately 5% of the total RNA of the cell, and it is short lifespan. It is having the short lifespan. This mRNA is having the short lifespan. That means it is degraded quickly after its function is over. When it gets decoded in the amino acid sequence in protein, after that it is being degraded, it is being broken down by the enzymes, RNases. So we can say that it lives in the cell for a short duration of time. After the completion of its function, that means protein synthesis, it gets degraded by the enzymes, RNases. And then we have two types of RNAs. One is called as monocistronic mRNA and polycistronic mRNA. We have two types of mRNAs. One is called as mRNA can be, this is what is mRNA can be monocistronic mRNA and polycistronic mRNA. Monocistronic mRNA and polycistronic mRNA. Polycistronic mRNA. So we have monocistronic mRNA and polycistronic mRNA. What this monocistronic mRNA is? Monocistronic mRNA. This monocistronic mRNA, this mRNA carries information for the formation of only one protein, and this mRNA is synthesized, it is transcribed from a single gene or single cistron. So this transcribed from, it is synthesized, it is transcribed from single gene or cistron. It is transcribed from single gene or single cistron, and this mRNA carries information for the formation of only one protein. Encodes, this mRNA, monocistronic mRNA, encodes only one protein. So it would be like this. And this monocistronic mRNA, these eukaryotic mRNAs are monocistronic mRNAs. So we have example, eukaryotic mRNAs are monocistronic mRNAs, eukaryotic mRNAs, or monocistronic mRNAs. It would be like this. For example, we have gene cistron, we have gene what we call as cistron. It synthesizes, it transcribes the mRNA. This will be monocistronic mRNA, monosystronic mRNA. Then this monocistronic mRNA synthesizes only one protein. It is only one protein. This is what is called as monocistronic mRNA. And these monocistronic mRNAs are found in eukaryotic cells. And these are characteristic of eukaryotic cells. So eukaryotic mRNAs are monocistronic mRNAs. So we have another type, what is called as polycistronic mRNA. Polycistronic mRNA. Polycistronic mRNA. This polycistronic mRNA, this mRNA encodes several, polycistronic mRNA encodes several proteins. And this polycistronic mRNA is transcribed from several cistrons, from several genes. So we use the term polycistronic. Poly, many, cistron means gene. That means this polycistronic mRNA is transcribed from several cistrons or from several genes. Transcribed from several genes or cistrons. Transcribed from several genes or cistrons. And polycistronic mRNA encodes several proteins. It encodes two or more than two proteins. It encodes several proteins. This polycistronic mRNA, it encodes two or more than two proteins. That is, it encodes several proteins. And we have bacterial mRNAs or polycistronic mRNAs or prokaryotic mRNAs or polycistronic mRNAs, prokaryotic mRNAs. Example of polycistronic mRNAs, prokaryotic mRNAs. Prokaryotic mRNAs or polycistronic mRNAs. So we have got this. If these are cistrons, these are genes, gene 1, gene 2, gene 3. These are cistrons. We have 3 cistrons, 3 genes. They will get transcribed into an mRNA. They will form a polycistronic mRNA. Polycistronic mRNA. And this polycistronic mRNA, then this polycistronic mRNA then synthesizes several proteins. So this is one. This polycistronic mRNA can synthesize 2 or more than 2 proteins, that is several proteins. So this is protein 1, 2 protein second, and protein third. That means 2 or more than 2 proteins are formed by the polycistronic mRNA. And this polycistronic mRNA, it is found in prokaryotic or bacterial cells. So it is a characteristic of bacterial cells. So prokaryotic mRNAs or polycistronic mRNAs. These polycistronic mRNAs are also called as polygenic mRNA. It's also called as polygenic mRNA. Polygenic mRNA. So what do you mean by polygenic mRNA? Polygenic mRNA is that mRNA that gets transcribed from several genes, from several cistrons. And monocistronic mRNA is also called as monogenic mRNA. Monogenic mRNA. Monogenic mRNA. Monogenic mRNA is that mRNA which gets transcribed from a single gene. So this is a monogenic mRNA. this monogenic mRNA, it is transcribed from a single gene, so we call it monogenic mRNA or monocistronic mRNA. This is polycistronic mRNA or polygenic mRNA. That means this mRNA gets transcribed from several genes or several cistrons. So what is called as monogenic mRNA, polygenic mRNA, monogenic mRNA, also called as monocistronic mRNA, polygenic mRNA, also called as polycistronic mRNA. This was what we mean by polygenic mRNA, polycistronic mRNA. Then we have another, what is the structure of mRNA? So we can here have the structure of the mRNA. We have the structure of mRNA. What is structure of mRNA? This mRNA, it is having, for example, this is an mRNA. This is an mRNA. This is mRNA. You consider that, and on this 5' end, 5', we have 7 methylated guanosine cap. We have 7 methylated guanosine cap. So it would be like this. If consider that this is an mRNA, and here we have what is called as 5' end, 5', 5' end, 5'. We know that at the 5' end we have phosphate group attached to the carbon 5th of the sugar. So at the 5' end of the mRNA, messenger RNA, what is present? There is 7 methylated guanosine cap. So we can have 5' end of mRNA. It is having 7 methylated guanosine cap. And this, at the 5' end of the mRNA, there is 7 methylated guanosine cap, and this 7 methylated guanosine cap, it is for the attachment of the ribosome. That means ribosome attachment site. Ribosome gets attached to this 7 methylated guanosine cap, ribosome attachment site. So we have this mRNA. We consider that this is a messenger RNA. This is the 5' end. That means phosphate group is attached to the carbon 5th of the sugar at this end. So we have 7 methylated guanosine cap. This is 7 methylated guanosine cap present at the 5' end. Then there is another region, what is called as leader region, or it is also called as a leading region. This leader region or leading region, it does not code for protein. It is known as untranslated region. It is called as untranslated region. So this leader region or leading region, it does not code for an amino acid. So it is called as untranslated region. So it would be like this. It would be here untranslated region. So it would represent what? Leader region. What is called as leader region. And then after this leader region, there is initiation codon, AUG or GUG. That would be here. We have got initiation codon, AUG or GUG. So initiation codon would be present initiation codon, and these initiation codons can be AUG or GUG, AUG or GUG. This AUG, it codes for methionine in eukaryotes and formyl methionine in prokaryotes, and this GUG, it codes for methionine when it is present at the initiation position. If it is present somewhere in the mRNA, then it codes for valine. Then we have initiation codon. This is what is called as initiation codon. Then this initiation codon is followed by a coding region, and ultimately at the end we have got what? Stop codon or termination codon. What is called as, we have then termination codons, termination codon, termination codon. Termination codons are also known as stop codons. We have UAA, UAG, UGA, UAA, UAG, UGA. Technically, these UAA, UAG, UGA are known as ochre. UAA is known ochre, UAG is known as amber codon, and UGA is technically known as opal codon. These are the German terms for the termination codon. So here we have termination codon. For example, UAA. This is termination codon. This termination codon is followed by another untranslated region, what is called as trailer region. Trailer region. This trailer region is also untranslated region. So we have leader region and this trailer region. They are untranslated regions. So this is followed by what region? It is followed by trailer region. Trailer region. This trailer region is untranslated region. It is untranslated region. That means it does not specify the amino acids. Then this trailer region is followed by approximately 200 adenosine residues. What is called as poly-A tail. What is called as poly-A tail. This poly tail, we have approximately 200 adenosine residues at the poly-A tail. We have poly-A tail. So this is at the 3' end, this is at the 3' end of the mRNA. So we have 5' end. It is having methylated guanosine cap, and at the 3' end we have poly-A tail. That means there are approximately 200 adenosine residues which are attached, which are present at the 3' end of the mRNA. Then we have this leader region, also known as leading region, plus trailer region. These are together known as what? Untranslated regions. These are untranslated regions. Because they do not specify the amino acid, we call them untranslated region. And this region which starts with the initiation codon and ends with the termination codon, it is known as coding region. It is known as coding region. So we have coding region. Coding region. This coding region, it is flanked by initiation codon on one side and termination codon or stop codon on the other side. It is also called as translational unit. This is called as translational unit. Translational unit. So this translational unit, it starts from AUG. It starts from initiation codon and terminates at the stop codon. This is what is called as translational unit. Translational unit is the coding region which starts with initiation codon, for example AUG, and then terminates with the stop codon, for example UAA, UAG, UGA. These are called as, this is a typical structure of what? Structure of mRNA, messenger RNA. This I told you that this Methylated guanosine cap, its function is the ribosome attachment site. That means ribosome get attached to the mRNA at this site, that is methylated guanosine cap. That means, for example, if this an mRNA, the ribosome will get attached here. That means at the 5' and 7 methylated guanosine cap. Then we have this UTR, untranslated regions. These untranslated regions, they increase the translational efficiency of mRNA. So they increase the translational efficiency of mRNA. Untranslated regions, they increase the efficiency of translation. They increase translational efficiency of mRNA. Then these untranslated regions, they also provide stability to the mRNA, as mRNA has to make a journey, as it has to travel from nucleus to the cytoplasm for protein synthesis. So we can say that these untranslated regions, they offer stability to mRNA, as it has to travel, as it has to make a journey from the nucleus to the cytoplasm for protein synthesis. Then we have another region that is called as poly-A tail. Poly-A tail. This poly-A tail, it is a tail of it is a sequence of approximately 200 to 300 adenosine residues which are present at the 3' end of the mRNA, and they also provide stability to the mRNA in the new environment. Stability of mRNA in new environment, in cytoplasm, I can see, as the nucleus is having one type of environment. When this mRNA travels goes to cytoplasm, there is another type of environment, and their mRNA is made stable by this poly-A tail. So we can say that this poly-A tail, it gives a sort of stability of mRNA to in its new environment in the cytoplasm. So we have discussed about the messenger RNA. I told you that messenger RNA, it is called as informational molecule, as it carries instructions from DNA to ribosomes for protein synthesis. Longest RNA, it is 5% of the total RNA of the cell. It is mRNA can be monocistronic mRNA, it can be polycistronic mRNA. Then we discussed the structure of this mRNA, and that means this mRNA can have at the 5' end and 3' end. We know that 5' end phosphate group is present and 3' end hydroxyl group is present, and methylated guanosine cap is present at the 5' end. This poly-A tail is present at the 3' end. Then we have got untranslated regions, we have got a trailer region, we have got a leading region, then coding region between. It is flanked by initiation codons and termination codons. And also we discussed what is the function of methylated guanosine cap. It helps in the attachment to the ribosome. This poly-A tail gives the stability in the new environment to the mRNA. Then AUG codon, we have got initiation codons AUG and GUG, and termination codons UA, UAG, UGA, and this was the structure of this typical mRNA. Thank you.